Hello guys and welcome back to Excelsior Academy. So today we'll be starting new topic, the cytoskeleton and the cell movement. So here, as you can see here also in this figure, you can see like it looks really very beautiful, with all those different colors and all. So these fluorescent tags which we see over here right now, like there are three proteins which is being getting tagged over here. So इस session में हम क्या देखेंगे, like how these cytoskeleton proteins, what are those cytoskeleton here, the actin filaments, intermediate filaments and the microtubules, right? So we are going to study about these. And if you remember, I mentioned the cytoskeleton as the highways which forms in the cell, जैसे कि रास्ता बनता है, so that these vesicles or the organelles can move from one place to another. Okay, and actin filaments are responsible for maintaining the 3D structure of your cell. So in this session, we'll see like how the cytoskeleton proteins are helping in maintaining the structure, उनका क्या function है, okay, and how they are made up of. Okay. The cytoskeleton, it consists of network of protein filaments which extends throughout the cytoplasm. The cytoskeleton, it provides structural framework for the cell, serving as scaffold that determines the cell shape as well as the positions of the organelles. Now, in addition to this structural role, it is also responsible for cell movement and transport of organelles and other structures, जैसे कि mitotic chromosomes through the cytoplasm. So we'll see that in the upcoming lectures also, like when we talk that जैसे कि आपका blood vessels हो गया, ठीक है, these are your blood vessels and this is the site where the wound has happened. Now we know that these WBCs, these the defensive cells, okay, so जैसे ये हो गए macrophages हो गए, so how from blood vessels, so we know that the blood vessels are made up of endothelial cells. Okay, now endothelial cells are there, so these macrophages, they will just go in between of these two cells, these two endothelial cells. अभी ऐसे, let us say that these are your endothelial cells. Now macrophages will move in between of this, right, and then it reaches its wound area. Now in order to go between of these two cells, it has to, the macrophage cell has to squeeze itself, right? ऐसे तो है नहीं कि वो इसको और इसको धक्का मारेगा. इन दोनों के बीच में जो interactions होता है, we'll talk later about it. So those are known as the tight junctions. Okay, so these tight junctions are broken and the macrophages squeezes itself and goes inside of between these two cells, और ऐसे बीच से बाहर निकलता है, ठीक है. Now for this you should understand that like so many filaments are there, और हमें मालूम है हमारा सेल ऐसे 3D स्ट्रक्चर में ऐसा रहता है कुछ. जस्ट इमेजिन उसको कितना स्क्वीज करना पड़ेगा अपने आप को, सो दैट बिटवीन टू सेल्स के वो बीच में से जाएगा, right? So when I say that the cell is getting squeezed, okay, what does it mean? It means that there are so many proteins which are getting polymerized, which is getting depolymerized. ठीक है, because suddenly there is a change in the 3D shape. आप 3D से उसको 2D बना के इसके बीच में से ला रहे हो, and then it has to move like this. ऐसे मूव करता हुआ वो अपने वुंड एरिया में जाएगा, ठीक है. सो जस्ट गेट द सिनेरियो ऑफ लाइक हाउ द सेल मूव्स एंड इन डिटेल, लाइक एग्जैक्टली इनसाइड ए सेल व्हाट इज हैपनिंग, विल डिस्कस अबाउट इट, ठीक है. Now see over here, so these are the three principal components of your cytoskeleton. So first one is the actin, which is in the red color. Microtubules, these are the green color, so this you see during the mitosis, and the intermediate filaments again in the green color. Okay, now are you able to understand the differences over here among all these three, among actin, microtubules and intermediate filaments? Akshaya, can you tell me which protein looks more abundant?
Microtubule.
It is the actin actually. No issue. See, the thing is, actin microtubules, if you see, these are like in this figure right now, you won't be able to understand, but once I show you the structure, once this topic gets over, I'll show you the video. There you can see that from one place to another place. Let's say that this is your cell, okay? This is how your microtubules are like this, ऐसे, single, sorry, not the last one, ऐसे. So if you remember when I was talking about the motor proteins, microtubules motor proteins, kinesin and dynein, so they carry the vesicles ऐसे, in this direction and in this direction, ठीक है? अब इनके बीच में imagine actin filaments ऐसे. So just imagine between the highways you have got so many small road. क्या बोलते हैं, कच्चा रास्ता जैसे नहीं बोलते हैं, तो वैसा. So there are like many are there. Okay. So you don't have any space in the nuclear, in the cell. You don't have any free space. जहां भी देखोगे बहुत सारे प्रोटीन है. अब इन सब के बीच में you have your mitochondria, peroxisomes, प्लांट सेल हुआ तो क्लोरोप्लास्ट, इन सब के बीच में ऐसे फिट है हो रखे हैं. ठीक है. This is where you have got the microtubules, active filaments as well as intermediate filaments. Intermediate filaments आपके कम रहते हैं, but you have got more of active filaments. ठीक है. We'll come to that, like how you can calculate all those things. Okay. So now first I'll talk about the actin filaments, then we'll go with the microtubules and the intermediate filaments. Okay. So first about the actin filaments. So in the actin filaments, they are the most abundant, and what you have to remember is that the diameter. Okay. S आपका actin है, so G actin is there. So G stands for globular actin. When it polymerizes, it becomes F actin. Okay. So from G actin, it becomes F actin, globular to filamentous. So filamentous means you have got G actin like this. When they start coming together like this, ऐसे long stretch बनता है, that becomes your filamentous. इसलिए उसको हम लोग F actin बोलते हैं. ठीक है. Please remember this 7 nanometer in diameter. So if you see later that G actin आपका सबसे छोटा है. ठीक है. So पहले आपका actin आता है डायमीटर में, उसके बाद IF आता है, उसके बाद माइक्रोट्यूबुल्स आता है. ठीक है. सो इन सभी में से माइक्रोट्यूबुल्स आपका सबसे बड़ा है. ओके. So within the cell, actin filaments, also known as microfilaments, they are organized into high order structures which forms the bundles also, as well as 3D with the properties of the semi-solid gel. ठीक है. अब see guys, imagine, okay, आपके पास पूरा सेल है, and when I say that it has got the properties of semi-solid gel, what does it mean? Like if you press it, what will happen? It can just change its structure. ठीक है. क्यों? Because of this actin filaments. ये actin filaments है जो 3D structure maintain कर रही है, but it is also making sure that it is flexible. आप जैसा चाहे उस shape को कर सकते हो. ठीक है. For example, अगर मैं बोल रही कि मेरा सेल right now ऐसा है, if I put a pressure over here, so the cell can become like this. Okay, so this is what I meant, like when I say that it has got the properties of semi-solid gels. Okay, did you get this point, like what I wanted to convey here? Krishita?
Yes, ma'am.
Okay. Now, the assembly and disassembly of active filaments, they're cross-linking into bundles and networks, and the other association with the cell structures like plasma membrane, they all are regulated by actin-binding proteins. So we have already discussed about this actin-binding proteins when we were talking about the ankyrin, band 3, glycophorin. तभी हमने ये सारे पढ़ लिए थे actin-binding proteins के बारे में. If you remember, spectrin, band 4.1, ये सब हमने तभी mention किया था. Okay, so again we'll gonna discuss all this thing. So they are the important components of the actin cytoskeleton. Now, actin filaments are particularly abundant beneath the plasma membrane, where they form network which provides mechanical support. Okay, determining the cell shape allows a movement of cell surface, thereby enabling cells to migrate, angle particles, and divide. Now, प्लीज रिमेम्बर, हम जब भी बोलते हैं ना कि ये सेल पर्टिकुलरली इट इज मैक्रोफेजेस इज ईटिंग द बैक्टीरिया एंड ऑल, सो कैसे रहता है, दिस इज योर बैक्टीरिया and let's say that this is your macrophages is there, right? So now the macrophages understand that this bacteria, I need to angle this thing, otherwise it will spread the infection, right? So for that, of course, it had these receptors present on the macrophages. It understands binding to the antigens which is released by the bacteria. Okay, so it binds and then what happens is that it starts changing its shape. कैसे? ऐसे से बनाना शुरू कर देगा. So what is happening? यहाँ के आपके एक्टिन फिलामेंट्स बन रहे हैं, ठीक है, और वो ऐसे बनते जा रहे हैं एंड यहाँ के टूट रहे हैं. So what is happening? This portion is leaving the area and here it is forming. So जिसकी वजह से आपका सेल इसके पास आ रहा है. So slowly it will come like this. तो ये एरिया छोड़ते हैं वो यहाँ आ गया, क्यों? बिकॉज यहाँ के सारे एक्टिव फिलामेंट्स तोड़ दिए इसने, यहाँ आ गया है एंड यहाँ पे बन रहा है एंड इवेंचुअली ये फ्यूज हो जाएगा यहाँ पे. थोड़ा सा आइडिया आया कैसे? I'll try to get the video. Okay, I'll search for the video. If I get it, then it will be more clear. So just imagine कि let us say this is a surface. Okay, ball कैसे roll होता है आपका? ऐसे से move करेगा, right? So ये portion आपका नीचे आ रहा है, ये portion आपका ऊपर जा रहा है, correct? So it means that here the new actin filaments are formed in order to push your cell like this, and here the actin filaments are getting broken down so that it can leave the surface. And plus ये अगर आपका cell है, ये आपका surface है, इसके बीच का interaction भी टूटेगा. So here inside these actin filaments are getting broken down. The interactions between the cell surface and here extracellular surface, it is getting broken. Okay, and here the upper portion, यहाँ पे आपका नया बन रहा है और यहाँ पे नए interactions बनेंगे. So that's how it rolls like this, and the movement of the cell is happening. Okay, did you get it? Little bit?
Yes, sir.
Yeah, good. Because this is really very interesting topic, the cytoskeleton and the cell movement. And once you get that clear picture, you will never forget in your life. So this is how in the electron micrograph your actin filaments you see. Okay. अब यहाँ पे देखो, it does not have any say proper connections. It's just randomly here it is there. So for me, for you right now, it looks like a random, right? But for the cell it is more proper, it is not random. Okay, it is very organized कि हाँ मुझे यहाँ पे active filament चाहिए. Why? Because this particular organelle is present here. I need to make sure that this organelle is present here and does not float away and go somewhere else. ठीक है. So हर एक ऑर्गेनल का अपना डेस्टिनेशन है. जैसे अभी हमने पढ़ा था हर एक प्रोटीन का अपना सॉर्टिंग टारगेटिंग डेस्टिनेशन है. In the same way the organelle also in a cell has got its proper location. ठीक है. Now, individual actin molecules are globular proteins of 375 amino acids. So G globular proteins is around 375 amino acids. Okay, so here each actin monomer, which is known as the G actin, it has got tight binding sites which mediate head-to-tail interactions with the other two actin monomers. Now from here you should understand that when from G actin, to from G actin to F it is becoming. It means what? The polymerization is happening. Correct? Now, when I say that one actin monomer has got tight binding for the other two actin monomers. So what does it mean? It means the starting material at the beginning, it needs at least 3 actin monomers to come together, and then onto top of it the other will join. ठीक है? और ऐसे ऐसे आपका filamentous बनेगा. So for starting it needs at least 3 actin monomers. Okay? Now, each monomer is rotated by 166 degrees, and because of which this finally the actin monomers appears to be like a helix, double stranded helix. तो वो थोड़ा सा ऐसे ट्विस्टेड दिखेगा. ठीक है? जस्ट बिकॉज ईच एक अगर ये एक्टिन है तो इसके 166 डिग्री में दूसरा है, फिर तीसरा है, फिर चौथा है, तो ऐसे ऐसे करते करते वो आपका डबल हेलिकल स्ट्रैंड दिखता है. ठीक है? So take away message from here, this point and this point. Now, since all active monomers are oriented in the same direction, अब कैसा है, यहाँ से आपका 3 है, राइट? इस पे आपने ऐड करना शुरू किया. Now, since everything is added in the one same direction, because of this now there is a polarity. So जो आपने जहाँ से ऐड किया है, that becomes your negative end, and where it is getting more addition of the G actin, that becomes your plus end. ठीक है? इतना क्लियर है? These points are clear, right? Because अभी आगे और थोड़ा complicated होता जाएगा, because we're going to add so many things to that. Now look at over here. So you have got the G acting like this. The moment it starts dimer and trimer जैसे ही बनता है, now it is going to start forming the filamentous actin. ठीक है? So minus end क्या हुआ? जहां पे आपने शुरू किया G actin add करना. Plus end where it will keep on adding the new G actin. Now, the question is, अगर ये minus end और plus end है, do they get stable or not? Are you getting the point here? Do they get stable or not? क्या हम हमेशा इसमें add करते रहते हैं? अब ये minus end है क्या? Does it mean that from here we start the depolymerization? So we have got those proteins, actin binding proteins, which stabilizes them, because when I say that inside the cell, and I say that there are so many interactions of actin filaments like this, right? So if the cell is not moving, if there is no movement, there is no migration, okay, then it has to be stable, right? So यहाँ पे मेरे पास capping proteins हैं, capping proteins, okay? Those are your actin binding capping proteins, which will bind at the minus end and at the plus end so that they become stable. ऐसे वो उसको bind कर लेगा. Okay, and this is the structure of your actin protein, and at the center you can see ATP binding is happening. Now let's see what is the role of ATP in the actin microfilaments. Okay, so we'll see when it is bound to ATP and when this gets hydrolysis क्या होता है. And it's very simple concept, हर जगह वही है. Like if it is ATP bound, then it results in the polymerization, and if the hydrolysis happens, okay, it becomes GADP, then there the depolymerization will start. Okay, the first step in actin polymerization, it is known as nucleation. So this is what, when I was just telling that where 3 G actin proteins are required. So that is nucleation, जहां पे आपका microfilament बनना शुरू होगा. ठीक है? So this is the nucleation, the first step in the actin polymerization. Now what is happening is that the actin, the G actin, which is bound to ATP, okay, they will go at the plus end, okay, and associate with the growing filament. So you can see here, G actin bound to the ATP, it is going at the plus end. Okay? Now, as soon as the hydrolysis of ATP is happening, GADP, sorry, GADP बन रहा है, what do we see? That it loses its affinity and starts depolymerizing. Again, there is exchange of ATP for ADP. क्या हो रहा है आपका G actin आ रहा है and again it is adding over here. So this is like a treadmill, यानी कि हम यहाँ add करते जा रहे हैं, यहाँ से हम minus करते जा रहे हैं. Again adding and again यहाँ से minus करना. And that's why it is known as the treadmilling of the actin filament polymerization. Is it clear? Okay. Now, within the cell, the assembly and the disassembly of actin filament is regulated by actin binding proteins. So guys, अभी यहाँ से हमें और भी proteins के नाम हैं जो हमें याद रखने हैं, and it is really very important because directly they ask. So some of these proteins initiate the formation of actin filaments or stimulate their growth, and the others are there which will stabilize, that is preventing the dissociation of actin monomers. So this is what I just mentioned, that there are stabilizing proteins known as actin binding capping proteins. Okay, and there are certain proteins which will help in the disassemble, यानी कि वो depolymerization के लिए help करेंगे. ठीक है. So the principal proteins which stimulate the initiation and elongation of these actin filaments are formin and ARP2/3 complex. Okay, so ARP is your actin related protein 2 and 3 complex. Please remember ये दो, formin and the ARP2/3 complex, where ARP stands for actin related protein. So the formins are the family of proteins which will bind ATP actin and nucleate the initial polymerization of actin monomers. So now we just saw that when nucleation is happening, it means G actin, okay? 3 molecules of G actins are coming together to start the actin polymerization. Now this is again, see, इनको भी एक base चाहिए कि कहाँ से हम शुरू करें. हवा में तो हो नहीं सकता. You need certain base onto which these 3 G actin molecules can sit and start nucleation, right? So that base is provided by the proteins formins. Okay. Now they then move along the growing filament, adding the new monomers to the plus end. Then they start adding like this. Okay. Now formins nucleate long unbranched actin filaments which make up the stress fibers, the contractile ring, filopodia, and the thin filaments of the muscles. Please remember, so in future अगर आपसे पूछा जाए that contractile ring हो गया या filopodia, as you know, that is, you know, जब भी हम pseudopodia, filopodia बोलते, that is a movement, the extensions of the cell. Right, ऐसे सेल है तो उसके लंबे से ऐसे एक्सटेंशंस निकलते किसी किसी सेल के. So that is because of the actin filaments. Now in that actin filaments also you need to understand that it is your long filaments which does not have got the branching. So ऐसे single सा fiber रहेगा ऐसे. So these are your actin stress fibers. Okay, so they are not branch. ऐसे ऐसे लंबे लंबे जो ऐसे रहेंगे, those are your actin stress fibers. Okay, now formins are associated with another actin binding protein known as profilin. Now this will bind actin monomers and stimulates the exchange of bound ADP for ATP. So just now we saw that in order to start adding the G actin, G bound ATP actin at the plus end, there is an exchange happening, right? ADP is getting exchanged with the ATP. Now that exchange is done by the profilin. Okay, so it means what? Profilin is helping in the polymerization. Okay, by increasing the concentration of your ATP bound actin. Did you get it? So abhi hum, let me summarize for you guys over here. Formin is it's forming the base onto which the 3 actin binds. Okay, profilin is helping in bringing the G actin bound to ATP. Okay, ARP2/3, this complex helps in the branching of the actin filaments. So right now what we have learned is about the long stretch of unbranched actin where formin, profilin is present. Okay, for branching we need ARP2/3 complex. Clear? Is this much clear? The functions of RF2/3 formin and profilin, guys? Yes, Tanvi, did you were you able to follow?
Yeah, ma'am.
Cool. Now look it over here. So we are going to discuss now formins RF2/3 and the profilin. Okay. So we have got a formin dimer. So if you remember, this is the base, okay, to which your ATP actin is coming. So 1, 2, and 3. So this is where the nucleation has started, okay, and onto which your more further actin filaments are joined. Ye clear hai, right? Now look at over here. So we have got the ADP actin. It is getting exchanged with the ATP with the help of the another protein called profilin. Okay. Now, so we have got the plus end where we are adding more ATP actin, right? And this is your minus end where the ATP hydrolysis is happening and it is your G with ADP. Okay. So this is your negative end. This is your plus end. Now, R2/3 complex is there, which goes and binds to your actin filaments. Now, what is happening over there? It is initiating the formation of the branch. So, we हम क्या देख रहे हैं? Though it is having here addition of your G actin is going on over here. Here also another branch is starting like this. So, at the end we see something like this. Okay. Now, one more thing what you have to remember here is that RF2/3 complex will only bind at the near your polymerizing G actin. Okay, that means it will be happening only near the plus end. Obviously, right? Because this is eventually gonna leave the association, right? Okay. Is this much clear? Clear to everyone? Yeah. Now the question should come over here, like how it is gonna leave when it is bound with the formin dimer. ये आपके base में है ना यहाँ पे अभी भी present. Correct? So when it is required that depolymerization should happen, then the base, the formin dimer will be removed. Okay, and then they start getting depolymerized. Okay, so here the RF2/3 proteins initiate growth of the branch actin filaments, which play key role in driving the cell movement at the plasma membrane. So this is how it is going to help actually in the movement. I'll try to explain with the diagram, and in future if I get a video, I'll show to you. Okay. So let's say that this is your cell surface onto which the particular cell is gonna move. Okay. तो just imagine एक ball है आपका और जब आप उसको roll करते हो तो कैसे होगा? When it is rolling in this direction, so these areas will come in contact over here, right? ये ऐसे move करेगा तो ये आपका यहाँ पे roll होके ये portion जो है ये आपका नीचे आएगा, correct? And these portions, ये वाला जो already contact में है वो ऊपर की तरफ आएगा, right? What you have to understand here is that the cell surface is having here the integral proteins and all, right? So these proteins, all the receptors are gonna make interactions with the surface, extracellular surface, ठीक है? And यहाँ पे जो already interactions है, उसको आप break करोगे. ये हो गया बाहर का, like outside the cell what is happening. Okay, now inside the cell, let's talk about inside the cell. So inside the cell, if this is there, so here you can see that आपके actin filaments ना ऐसे ऐसे spreaded है, ठीक है? Now this portion is going to come here, right? ये portion को मुझे यहाँ पे लाना है, push करना है, cell को move कराना है. So what will happen? ये branch करता हुआ आपका actin filaments ऐसे ऐसे इसको push करेगा, okay? यहाँ से. And what is happening over here? ये आपका गायब होने लग जाएगा, ऐसे. Okay? So eventually what is happening then? When I'm making here the actin filaments, this portion will come down. And here there is no actin. Okay. So what will happen? ये जो पोर्शन आपका लीव कर देगा, छोड़ देगा, एंड ये पोर्शन आपका नीचे आ जाएगा. सो इवेंचुअली दिस इज कमिंग डाउन. सो अभी ये नीचे आ गया है. नेक्स्ट क्या होगा? यहाँ पे आपके एक्टिन्स बनेंगे और ये गायब हो जाएंगे, फिर ये आएगा नीचे. सो दिस इज लाइक हाउ अ बॉल इज रोलिंग. तो आपके ऐसे ऐसे एक्टिन फिलामेंट्स बनते जाएंगे, पीछे वाले टूटते जाएंगे, एंड दैट्स हाउ योर सेल इज मूविंग. आया थोड़ा सा आइडिया? Yes, 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 good. It's really important to get that idea. Okay, so this is how the actin filaments looks like. So when I was talking about the stress fibers, right? So those are the bundles. ऐसे straight रहेंगे आपके. And of course, with each actin, you are going to have the cross-linking proteins present. ठीक है? Otherwise, what will happen? The bundles खुल जाएंगे आपके, right? वो इधर उधर चले जाएंगे, scatter हो जाएंगे. Whenever we have bundles, what we do? We tie it, right, with some rope or whatever. In the case of the cell, these stress fibers, they are actually interconnected with each other with the help of the again actin binding proteins. Okay, now here again, if suppose network बनाना है, so as I was see, this is how ऐसे जो network दिखता है, ये हो गया आपका stress fibers single. यहाँ पे आपका network चालू है actin का. This is your stress fibers. यहाँ पे आपका network है. So for us, it looks like that is, you know, random एकदम it's like zigzag. कहीं से भी कुछ भी cross connection बने हुए, right? But for the cell, it is all planned. It's not random. It is like organized. So here you can see so many actin filaments are going here, and each one is actually being connected. Can you see? So with the help of the cross-linking proteins, everything is being connected. ऐसा नहीं है कि that it is, you know, randomly associated. Now there is another actin binding proteins which remodel or modify, okay, rather than stabilizing, and that is your coffulin. Okay, so actin binding protein coffulin, now which is going to severs the actin filaments. So severs the actin filaments मतलब it is going to break the filament. Profilin क्या कर रहा था आपका? It was helping in addition of your G actin bound to ATP, correct? Coffulin क्या करेगा? It is gonna break it. Okay, because of which it is going to generate new ends of the filaments, which are then accessible either for the depolymerization from the minus end or for the growth of the new actin monomers to their plus end. So here, see, ये आपका minus end हो गया, ये आपका plus end हो गया. Now let's say that there were these stabilizing proteins because of which minus end से आपका depolymerization नहीं हो रहा था और plus end में आपका more addition नहीं हो रहा था. ठीक है? Now let's say that there is a coffulin coming. Coffulin आएगा, वो क्या करेगा? Actin filaments को बीच में तोड़ देगा. Okay, so what will happen because of this? There will be a minus end generated and the plus end generated. So plus end में क्या होगा? You can have more addition of your G actin bound to ATP, or at the minus end, the G actin bound to ADP will start getting depolymerized. Okay, so now I hope the functions of formants is clear. Profilin is clear. ARP 2/3 is clear. And then last we have talked about coffulin, right? I hope the functions of all the four is clear. Formin, it forms a dimer base onto which your nucleation initiates, right? Profilin helps in binding of your GATP in the plus end, like it keeps on helping in the addition of your plus end में add करता है, right? It is helping in the polymerization. ARP 2/3 is helping in the branching. Coffulin is helping in the, it breaks the actin filament so that the polymerization and depolymerization both can happen. Guys, is it clear? Akshaya?
Yes, ma'am.
Okay. So this is a video let's see how our understanding is. Okay, so just देखो कि कैसे आपका imagination and in reality how it is happening, it is matching or not. Okay, so let's see this video. There is no sound. Okay, now it has become ADP actin. So yellow color they have shown as your ATP bound actin. So the moment 3G actin will come together, the nucleation will start. So you can see like how plus enders and minus enders form. Minus end say depolymerization is happening. This is your actin treadmilling. Like, one end it's keep on adding, the another end it's keep on depolymerizing. See, in between the coffin is coming and it is breaking the filaments. Okay. Yes. Guys, any doubts in this topic today?